Today we're going to be looking at biological catalysts, and we're going to link this to our lessons that we have previously completed on the digestive enzymes. So you need to be able to keep referring back to the theory on digestive enzymes and link it now to the theory of enzymes as a whole as biological catalysts. So we're going to look in a bit more detail now on the mechanisms of how these biological catalysts work. Okay, so the first objective is that we need to define the word enzyme, and you will need to have a good understanding of how to define this word enzyme, because you can be asked this in a number of ways. And the second part is to explore the mechanism by which enzymes work. So we'll come on to factors that affect enzyme activity, but before we do that, we need to explore the mechanism by which enzymes work. We're talking about enzymes and enzyme activity. We are essentially focusing our attention on the rate of a chemical reaction. So the reason we use enzymes, or why enzymes are important for living organisms, is to do with this concept of a rate of a chemical reaction. Now, enzymes belong to a larger group of chemicals known as catalysts. So first we have to understand that enzymes belong to a larger group of chemicals enzymes belong to a larger group of chemicals known as catalysts. Okay, so can you just take that down first? Enzymes belong to a larger group of chemicals known as catalysts. You can have organic catalysts and inorganic catalysts. Inorganic catalysts are important in industry, and also you can have biological catalysts in industry as well. For our focus, we are focusing on biological catalysts, which are known as enzymes. So the first distinguishing point is enzymes belong to a large group of chemicals known as catalysts, and you should be aware that you can have organic catalysts, i.e. enzymes, biological catalysts, or you can have inorganic catalysts. So we know enzymes are catalysts, but what is a catalyst? So a catalyst is a chemical that will speed up the rate of a reaction, will speed up a chemical reaction, but will not be used up in the reaction itself. This means that you can reuse a catalyst over and over again. So let's put this into words. Okay, a catalyst will speed up a chemical reaction, but you can say however, it is not used up in the reaction. So a catalyst will speed up a chemical reaction, however it is not used up in the reaction itself. Okay, now this allows and this next point is important, Therefore, catalysts can be used over and over again. Now, I've kept this definition to catalysts, but based on our first point, you should understand that this is also the concept for an enzyme, because enzymes are essentially biological catalysts. Now, the reason I've given you a generic definition for catalysts as well, because when you start to look in certain topics in chemistry, we will come across this word catalyst again. So a catalyst will speed up a chemical reaction, however, it is not used up in the reaction. Therefore, catalysts can be used over and over again. We're going to look at the enzyme lock and key mechanism. So on the screen you can see the enzyme and the substrate represented. Since we have an enzyme, we have to identify the active site, and the active site is here. So let's say this is the active site. So this is where the binding of the enzyme and substrate is going to happen. And notice that the active site needs to somehow combine with the substrate in a correct manner for a reaction to take place. Now we've identified the enzyme and the active site. Let us see how the substrate will interact with the enzyme. So if we were to move this enzyme over to the substrate, okay, and we were to see how it fits in, you can see that the shape of the substrate to the active site is complementary. And what happens here is that by the substrate having a specific shape for the active site means that biological enzymes are specific in nature for their chemical reaction. So this is just a crude example of how the enzyme has a specific active shape where it will allow the substrate to join with it. This is called the lock and key mechanism, because if this shape was not a specific shape, if the substrate did not have a specific shape, and also if the active site was not specific in its shape, we could not have this binding of the enzyme and substrate. So the enzyme and the substrate would not be able to bind if they did not have a specific shape. So certain enzymes, specific enzymes, so amylase, let's say, has this type of shape. The amylase enzyme would have this type of active shape where the substrate could bind to, where the carbohydrate could bind to. However, protease would have another type of shape to which the actual substrate could bind to. So we say that enzyme substrate reactions undergo a lock and key mechanism type of reaction, whereby which specific active site shapes allow for specific specific substrates to bind and allow a reaction to proceed. Once the substrate has bound to the enzyme active site, a chemical reaction can take place and a new product is formed. So the substrate is changed, but notice that the enzyme has not changed. So once there has been a binding of the substrate and enzyme via a specific active site found on the enzyme, the substrate is changed and the enzyme remains unchanged once the reaction has taken place. So this is one of the key main points of enzyme reactions, that the enzyme will remain unchanged once the reaction has proceeded and a product has been formed. Substrate will change, but enzyme will not change. Once the reaction has happened, a new product is formed. So the substrate is turned into a product. Now, once this product has been formed, it is released by the enzyme. So it is released by the enzyme, and as you can see, we have a new product being formed and we have the enzyme unchanged. So the product is released and it goes away from the enzyme. Now, why is this important? Because what we have here now is that the active site is now free. So free active site. Okay, a new product has been formed and the enzyme is unchanged, and now there's a free active site. So this allows for any substrate to come in now and for another reaction to happen. Okay, so we've had one substrate come in already, bind to the active site of the enzyme because of its specific shape. A reaction has taken place, a product was formed, the product was released, and the enzyme remained unchanged. So because the enzyme has remained unchanged, it allows the enzyme to break down another substrate or convert another substrate into a product. Now, I've shown the product being in a whole piece. However, products can be in separate pieces as well. So you can have enzymes breaking down substrates. Here, a separate product was formed and it stayed intact. However, enzymes can break down substrates into different products. So now we're going to look at various lock and key mechanisms. I'm going to show you a variety of diagrams that show enzymes in various shapes and various substrates as well that have a corresponding complementary bonding. Now, the reason I'm doing this is because for you to actually appreciate that there are various enzymes, they are specific in terms of their shape and ability to bind to substrate. And what I also want you to look out for is how the products are being formed. So enzymes can break apart substrates into multiple products, or enzymes can be joining two substrate molecules to form a product. So what we're going to do now is look at the various ways in which enzymes can work with substrates to form products or product. So let's take a look now. Here you can see a standard lock and key mechanism being illustrated. It's showing that the substrate here, if we take this as our first point, it shows that the substrate enters into the active site. Enzyme substrate complex is formed. So we have an enzyme substrate complex which is formed, and then a product is created, and in this case you can see that the substrate has been broken down and two new products are essentially formed as a result. And just notice that in the final stage, so if this is 2 and this is 3, you have something which is also mentioned here as an enzyme product complex that is briefly formed as well. Now notice that in each instance, even the enzyme substrate complex is temporary and the enzyme product complex is also temporary, briefly formed. So these are just points in which they are describing how a substrate will enter a specific active site shape and the mechanism by which we get products. Now these reactions of substrate coming in and binding and then a product being formed, they are not slow processes. They happen very fast. Depending on, most enzymes are able to do this at a quick rate, and we'll talk about this in more detail, because biological reactions, for living organisms to thrive, we need reactions to happen at a very fast rate, and enzymes aid in this. Here you can see a substrate approaching a specific enzyme with active sites, multiple active sites, and also notice that the enzyme changes its shape ever so slightly as the substrate comes in. This is because although we say that enzyme substrate complexes are similar to something like a lock and key mechanism, where they are highly specific in terms of their shape and their complementary shapes, there is some room for movement in terms of the active sites can ever so slightly change their shape to accommodate certain substrates, and the substrate can also in certain instances change their shape very slightly to ensure a good binding. So although enzymes are considered to have a specific active site shape which is highly specific for the substrate, there is a theory that has also been put forward where, or a hypothesis that's been put forward, where there's a changing, an ever so slight change of the enzyme active site. Now this is known as the induced fit model, where the enzyme's active site can change very slightly, and that's what you're seeing here. Notice here that the enzyme substrate complex is formed by two substrates that come together and form an enzyme substrate complex. This is different from before, where we had one substrate coming into the active site. So enzymes can also work in this manner as well. So this would be another type of enzyme that is joining two substrates together to form a specific product in this case. So two substrates come together and a product is released at the end. And notice again you have your enzyme substrate complex and then you have an enzyme product complex. Notice this region here is different from, it is no longer found at this point here. So you can see that we have another type of complex being present, and an enzyme substrate complex is held together by specific bonds as well at the active site. So for this all to occur, bonds are made and bonds are released. So this is why we have this word enzyme substrate complex, enzyme product complex, for example, as you see it represented here. Specific bonds are formed to hold these complexes in place. Specific bonds are formed. Here you can see another type of enzyme shape, or another type of active site, and what you can see from here is that here two products are formed. So it goes like this. So this is the order in which this particular diagram is representing: substrate coming into the enzyme substrate complex being formed, and then the products being released eventually. So this is another way in which a substrate and an enzyme can interact. Okay, now here you can see an enzyme substrate interaction by which two products are being formed, and you can clearly see how the two products are different from the substrate itself. Now you can also see here the diagram is representing via that big blue arrow at the bottom, that the enzyme is ready to use again. Now I want you to take a look at this diagram, because I'm going to show you one more diagram after this, and you may choose to draw this diagram or the one that's coming next. So just study this diagram carefully. If it makes sense for you in terms of if it concisely allows you to see what's going on and allows you to explain an enzyme substrate complex in a 4-mark to 6-mark question, i.e. you're able to get enough information from this diagram, then take this diagram down after comparing it to the next diagram. This diagram essentially gives you the lock and key mechanism. The next diagram, which I will show you the lock and key mechanism in addition to the induced fit model. So study this diagram carefully and compare it to the next one, and whichever one you feel that will help you understand and be able to write an extended answer question on, take that diagram down. But you must take one of the two diagrams in your notes. So again, this is a simple lock and key mechanism displayed here. The key point here is that the substrate fits into the active site, and I would say at this point here, maybe write down enzyme substrate complex. Okay, as we have been seeing previously in the other diagrams. So here you have another diagram, and here you can see the lock and key hypothesis and the induced fit model next to each other. And what I want you to do is to compare this to the diagram that was just previously shown, and I want you to draw one of the two. Now this one has a bit more information here, so I would recommend taking this one down, as it shows for the lock and key hypothesis that a product is formed and the products are different from the substrate, but the enzyme is left unchanged. The only addition to the lock and key hypothesis diagram, I would say, is that make sure there's an arrow saying that this enzyme is now available for another substrate to bind to it. So that would make the lock and key hypothesis diagram a bit more, let's say, completed in a better way. Then you've got the induced fit model below, and notice here, let's just take a look at the active sites in a bit more detail. So what you can see here is that the active sites have changed very slightly to accommodate the substrate. Notice this region here and this region here is very different. It is different. Okay, so the shape has changed, and this has happened to accommodate the substrate in a better manner. So what I will say is that decide which diagram you wish to take down. I would recommend this diagram here, as it gives a bit more detail, but as I said, for the lock and key hypothesis, maybe put an arrow saying that like this to indicate that the enzyme is available for use by another substrate. So just take now maybe let's say 5 minutes should be fine. If you take 5 minutes now to draw your lock and key mechanisms for your enzyme, so by diagram you choose of your choosing, take 5 minutes to draw the lock and key mechanism and label it correctly. So let's say task will give this 5 minutes. Here you can see the 3D structure of enzymes. Now enzymes are essentially globular proteins. So you need to understand this term globular. Now, a globular protein has a 3D structure, a three-dimensional structure, and because of the coiling nature of enzymes, these structures that are formed are highly specific, and the folds and bends that you see in this globular protein allow for it to bind to a specific substrate and gives rise to the specialist nature of enzymes. Now, as we've looked at previously, when we were looking at protein folding or protein structures, that the amino acids and the resulting chains that are formed are able to fold in a particular way. And this is an example of a protease enzyme, a type of protease enzyme. And you won't need to know in detail about the 3D structure or the globular nature of it. However, this gives you an idea of what the actual what we're talking about when we say a 3D globular protein, and how this gives rise to the specialist nature of enzymes. So we were looking at previously just simple model diagrams of enzymes. However, the enzymes look something like this, where you're seeing multiple folding layers or coiled forms of the protein, and this is what the enzyme actually looks like. And this is one example. This is a protease enzyme, and this is the nature, or this is how they look in terms of when a protein has been folded and how they would look in real life. So now we're just going to summarize enzyme mechanisms. So we've looked at them in terms of the various mechanisms that can be attributed to how enzymes work, and now we're just going to summarize what we've actually looked at. Okay, so enzymes are proteins which can be defined as biological catalysts. So enzymes are proteins. Okay, so can you just underline the word proteins, which can be, let's say, defined as biological catalysts. Okay, so that's an important word as well. Right, enzymes are proteins which can be defined as biological catalysts. All metabolic reactions that take place in a living organism is catalyzed by enzymes. So can we put down almost all chemical reactions well, let's say instead of chemical, let's use a better word, almost all metabolic reactions, almost all metabolic reactions that take place in a living organism okay, so all metabolic reactions that take place in a living organism are catalyzed by an enzyme. Okay, so almost all metabolic reactions that take place in a living organism are catalyzed by an enzyme. Next point. we'll say that enzymes are globular proteins. Therefore, enzymes are molecules that are coiled into precise 3D shapes. So can you take down enzymes are globular. Okay, so globular, can you just underline the word globular proteins. Okay, so enzymes are globular proteins. Thus, they are coiled into, let's say, specific three-dimensional shapes. Okay, so enzymes are globular proteins, thus they are coiled into specific three-dimensional shapes. Their globular nature gives them their three-dimensional specific shape. Now the next point is, let's put it in another color, let's make it a bit more visible. The shape of the active site allows a substrate to fit perfectly, and this is known as a lock and key mechanism. So we'll say the shape of the active site the shape of the active site allows the substrate the shape of the active site allows the substrate to fit perfectly forming an enzyme substrate complex. Okay, so could you just take down how the active site allows for the perfect fitting of the substrate, forming an enzyme substrate complex? Can you finish this off by saying this is known as the lock and key theory? Okay, due to the specific nature of the active site and the way in which the binding occurs, this is why we call it the lock and key theory. Just a final point on the enzyme substrate complex, just a sentence that you can add a bit more. Can you write down that the substrate is held in place via temporary bonds. Okay, so the substrate is held in place in the active site via temporary bonds. Okay, let's say with the active site to make it clear. With the active site. So this is how the enzyme substrate complex is formed. Okay, so this is known as enzyme substrate complex. The formation of temporary bonds holds the substrate in place. Now, when we talk about the enzyme mechanisms involved and how we are forming a product, just be aware from the diagrams that we looked at previously that an enzyme can join small molecules together as well as breaking apart large substrate molecules. So can you just write down enzymes are able to join small substrate molecules small substrate molecules to form a product. Okay, and enzymes can also break apart substrates into products. So we have different actions that an enzyme can potentially take, and again this is due to the specific folding that a protein has undergone. Now what we're going to do is we're going to look at the different ways in which biological enzymes can work in your body, and these are in metabolic reactions. Now we came across the word metabolic reactions just a few minutes ago, and we're going to look at metabolic reactions and enzyme activity in a little bit more detail now. So as we move on to metabolic reactions, can we just put this sort of reminder about enzymes? Enzymes do not change a chemical reaction. They do not change the nature of the chemical reaction. They just allow the reaction to happen faster. They just make it happen faster. Okay, so enzymes do not change a chemical reaction, they do not change the nature of the chemical reaction. They just make the reaction happen faster. Now, metabolism is a sum of all the reactions in a cell or body. Okay, so can you write down that metabolism, let's put it in a different color to make it more. Can you write down metabolism? Metabolism is the sum of all the reactions in a cell or body. Certain enzymes will be having their mode of action within the cell. Certain enzymes have their mode of action outside the cell. So you have intracellular enzymes that are found in the body, and also you have enzymes that are not located to any individual cell inside, but are found outside the cell. So metabolism is a sum of all the reactions in a cell or body. There are essentially three types of metabolic reactions that we are going to look at. We're going to look at metabolic reactions where we build large molecules from lots of smaller ones. Another type of metabolic reaction is changing one molecule into another. And the third type of metabolic reaction is where we break down large molecules into smaller ones. So let's take a look at these three types of metabolic reactions. So the first one, building large molecules, okay, from lots of smaller ones. Okay, building large molecules from lots of smaller molecules. And an example of this, so let's have some examples of this type of metabolic reaction. Okay, so, and I want you to just take this down so you know this is a type of metabolic reaction and how it's coming about. Building starch. Okay, that's one type. Now I'm just going to put building, I'm not going to write building each time, but so you get the idea. When we make glycogen or build glycogen, okay, and cellulose, okay, and these ones, so these three molecules, sorry, these three, let's say larger molecules, are made from, so I'm not going to tell you, I want you to just pause the video here and tell me what molecule, what smaller molecule would we use to build starch, glycogen or cellulose. So these three larger molecules, starch, glycogen and cellulose, could you just pause the video and tell me and write down in your notes what smaller molecule we could use to build up starch, glycogen or cellulose? And could you type back the answer to this on Teams to me so I know that, so I can just check that you are following this correctly. Can you just write down the name of the smaller molecule that could be used to build starch, glycogen or cellulose? Okay, you should have the answer as being glucose. Okay, so glucose, the glucose molecule, would be used to build up starch, glycogen or cellulose. Now, another example of a metabolic reaction where we build large molecules is lipids. Okay, lipids from fatty acids. Okay, lipids from fatty acids. And if I was to say proteins here, can you tell, if I leave this blank, I want you to put down in your notes what proteins would be built up from. And could you, if you know the answer to that also, if you're confident, can you type that back on Teams, what proteins are built from? And to make sure that, no, we'll leave it there. We'll leave it there. And you can also note down in your notes that combining carbon dioxide and water, we can make glucose. So glucose can be made if we look at it even further, glucose is made by plants, and the glucose molecule is made by plants when they combine carbon dioxide and water, and they use glucose and nitrate ions to make amino acids. So even this glucose here, which we consider it to be the smaller molecule, when we think about plants, they are the organisms that make the glucose, and they make the glucose from combining carbon dioxide and water, and then they also use the glucose to make amino acids via combining it with nitrate ions. So plants can make glucose, and they can also make use of glucose by combining it to nitrate ions to make amino acids. So the next type of metabolic reaction we can have is changing one molecule, okay, into another. So we can change one molecule into another, and this is a type of metabolic reaction. Examples include, okay, turning a simple sugar, let's say one simple sugar, turning one simple sugar into another. Now we looked at the types of sugars earlier, so an example of this would be glucose to fructose. Turning one simple sugar into another, glucose to fructose. And we can also have an example where we're looking at turning one amino acid to another, and we looked at the various types of simple amino acids that we can have. So converting one amino acid into another. Now the reason I'm stressing these points here and asking you to take this down is because there have been exam questions that will give you a list of, let's say, reactions that are taking place and ask you to identify. I have seen ones where they ask you to identify which is a metabolic reaction, or explain why this is a metabolic reaction. And they can be just simple 2-mark questions, 1-2-mark questions, where you have to either identify or just give a quick brief 2-mark answer. So you should be aware of the different types of metabolic reactions just to be able to identify them. So this is the second type. Now we'll move on to the third type of metabolic reaction. The third type of metabolic reaction in which an enzyme catalyzes, i.e. speeds up the specific reaction, is breaking down large molecules into smaller ones. So the third metabolic reaction that we're looking at is breaking down large molecules into smaller ones. Into smaller molecules. So this is the third type of metabolic reaction. Okay, e.g. examples. So let's look at some examples. Okay, we can have breaking down carbohydrates, lipids and proteins into their building blocks. Okay, so carbohydrates would be broken down into their simple sugars. Lipids, we know that lipids are made from fatty acids and glycerol. So if you were to break down a lipid, you could get it down to its building blocks, and proteins into their constituents. So examples include carbohydrates, lipids, and proteins. Notice how we can build larger molecules, and also we can break them down. The same molecules can be broken down. So this is one example of where metabolism is being used. Now, this breaking down of carbohydrates, lipids, and proteins, okay, occurs during digestion. So this metabolic pathway occurs during digestion. Now, you have the breaking down of glucose. So glucose can be broken down. Breaking down of glucose occurs in cellular respiration. Now, this is also something that is asked quite frequently in exam questions to identify the metabolic reaction, and cellular respiration is one example, because we break down glucose to provide us with the energy that cells need to survive. Now, the final metabolic pathway here that I want you to take down is that breaking down excess amino acids to form urea. Okay, so the final type of metabolism for breaking down large molecules, can you take down that we break down excess amino acids to form urea. Okay, so the breakdown of excess amino acids to form urea is a metabolic pathway. Now I'll repeat again, these enzymes which are involved in metabolic reactions, they are catalyzing, speeding up these metabolic reactions. And we've looked at 3 types of metabolic reactions, and I've given you some examples. So you will need to know the types of metabolic reactions with examples. Once you've completed your notes, can you now complete the task on Teams? The questions that I've uploaded on Teams, can you start working your way through those questions that have been uploaded there? And can you submit your answers back to me on Teams?